And these vaccines collectively had really remarkable successes at improving human health. So probably everyone knows we succeeded in eradicating smallpox globally, and that had real consequences. So it was a horrible, devastating disease, caused lots of morbidity. In the sort of same vein, we've now driven polio to the brink of extinction through the effective use of vaccines. And obviously, at least within North America, we're also able to pretty effectively control things like measles, mumps, rubella with routine vaccination. But despite these successes, we really have still a lot of challenges that we've been unable to overcome. And one of those challenges, this kind of a perennial challenge with vaccination, is vaccinating a sufficient proportion of the population to eradicate an infectious disease. And one way to understand this is a classical mathematical result, okay? And that mathematical result shown here tells us that the proportion rho of the population that we have to vaccinate in order to eradicate an infectious disease is equal to 1 minus 1 over this quantity R naught. And R naught is really a measure of the number of new infections produced by a single infection. Okay, so if that R naught quantity is greater than 1, each infected individual on average infects more than one other individual, and the infectious disease is spreading through the population. If it's less than 1, it's on its way towards extinction. Okay? Now, an important consequence of this result is that as the R naught of an infectious disease increases, so too does the proportion of the population you have to vaccinate. Okay? And that can be very challenging under some circumstances. Okay? So it's achievable in a place like North America where we have excellent public health infrastructure. You can reach these levels of, say, 70, 80% of people being vaccinated. It's much harder in areas of the world that have less well-developed public health infrastructure. And an area where it's becoming increasingly important is if we're to try to use vaccines to eliminate infectious disease from wild animal reservoir populations. And in those cases, it's non-trivial to vaccinate even a modest proportion of a population of wild animals. Now, another sort of related challenge is vaccinating quickly enough. So you can imagine a scenario where, for instance, a new pandemic is about to spread, right? Or we see an emerging infectious disease somewhere. We want to deploy a vaccine rapidly enough to stop that infectious disease. Well, it turns out this is actually a lot harder to do than you might imagine at first pass. So what I'm showing you here is just some simple results that show how long it takes on the y-axis here in terms of days to vaccinate a population and protect it from diseases with different R naughts. Now, this figure is predicated on a vaccination rate that's the equivalent of what we're able to achieve in the United States for seasonal influenza. So in principle, you could raise that up some, but probably not orders of magnitude. And what you see is that for something like influenza, it takes around 220, 230 days to vaccinate the population sufficiently to protect it. For something like SARS, you're almost up to 400 days. If something like smallpox were to escape from the lab, you're up to, it's totally impractical. You are never going to vaccinate the population fast enough. Even if you already have the supplies manufactured, you're not going to succeed. Okay? So those are two problems we've got. One way that we could potentially surmount some of these difficulties is by using a transmissible vaccine. So the idea here is you have a vaccine that's replication competent and able to actually transmit from host to host. So if you can do this, then if we say, for instance, directly vaccinate those two individuals, so that took 2 doses of vaccine, it took us 1 point of time, and we get 2 bonuses, right? And then maybe even another bonus. So that can improve our depth of coverage given a finite amount of vaccine, which is what I'm showing here. You could imagine that, say you need to get above this threshold to eradicate influenza, and with a traditional vaccine you can only get here. Maybe vaccine transmission would bump you up above the eradication threshold. Similarly, you could imagine, if we're thinking about the time required to protect a population, having vaccine transmission, that could lower that time to protection of the population. Okay? So those are some potential benefits we might realize. Now, what's really interesting about this, and which was part of how I got pulled into this project, is this isn't science fiction. So we actually have transmissible vaccines. Many vaccines are replication competent already, and any replication competent vaccine can, in principle, transmit. And the fact is, if you actually look through the data, all of these vaccines that we use routinely have documented cases of transmission. Now, the most striking example of that is the oral polio vaccine, where extensive transmission is well documented, and that has both positive and negative consequences that I'm going to come back to later in the talk when I talk about some of the consequences of evolution. The other thing that makes this even more important to understand now is that people are actually engineering these transmissible vaccines for wildlife populations. The primary goal is to develop a tool that allows infectious diseases of humans to be eradicated from wildlife reservoir populations. Okay? So things that are currently underway are a vaccine against Ebola in fruit bats and hantavirus in deer mice. There were also earlier efforts to develop a transmissible vaccine against myxoma in rabbits, and that's the only one that I know of that has been intentionally developed and was actually taken all the way through to field trials and shown to work. So this isn't science fiction. We've already developed transmissible vaccines. There's two ways in which this can be done. One of them is through traditional attenuation. Here the idea is pretty straightforward. You take the wild type pathogen and you just passage it in the lab under unnatural conditions. For instance, maybe you passage it from hamster cell to hamster cell under cold conditions. And as a consequence, of course, you accumulate mutations that adapt it to that environment, but are negative in the original host. And what that leads to is attenuation. So the vaccine still inspires immunity in the host, but it doesn't have very high virulence or no virulence at all. Okay? So an example of this is the oral polio vaccine. That's the classic example of a transmissible vaccine developed through attenuation. The other more modern way in which transmissible vaccines are being developed on purpose is through recombination. The idea here is you take some vector, some transmissible but innocuous organism that already occurs, for instance, within the host population. And you take a gene from the pathogen genome, something that has known antigenic activity in the host. You insert that into the you insert that into the vector genome, and that is your transmissible vaccine. This is how the myxoma virus vaccine in the rabbits was developed, and it is how current efforts are proceeding to develop the vaccine against Ebola in fruit bats and hantavirus in deer mice. So what I see here is something like this. We've got this new technology emerging that in principle sounds both promising and somewhat frightening, and yet we don't have any mathematical framework or theory to really understand the conditions under which a transmissible vaccine will be effective. For instance, how effective is it going to be? Is it going to be just a little bit effective, in which case maybe we shouldn't be pursuing this, given the potential risks? Or is it going to be hugely effective? The other thing we don't really understand or know how to manage yet is the potential for evolution in these transmissible vaccines. So what I want to do today is tell you about work we've been doing in my lab, trying to develop at least the sort of start, the very beginnings of a mathematical theory for understanding, for quantifying the potential benefits of a transmissible vaccine and also the consequences of evolution and how those might be managed. So the questions I want to target today specifically are, first, now I know why you're laughing. Okay, it's always three. That's from John. So could vaccine transmission facilitate disease eradication? So it's obvious vaccine transmission is going to be probably beneficial in some way, but what we don't have are any real numbers on this. Is it a little bit or, a massive amount? Second thing I want to talk about is whether or not vaccine transmission could be beneficial in terms of the rate at which we can protect a population. And then lastly, really get into some of the nitty-gritty of what we expect to happen when these things evolve. Because once you've got a live replication competent vaccine, it's subject to evolution. The modeling framework I'm going to use throughout builds off this general classical framework used in infectious disease. And really what we've done is we've just taken classical SIR type models of vaccination and allowed the vaccine to transmit. okay? So the basic idea with all the models I'm going to talk about today is that we've got susceptible hosts, we've got pathogen-infected hosts, vaccine-infected hosts, and resistant hosts. The transitions we're going to model are that susceptible hosts get directly vaccinated at some rate sigma, and they turn into vaccine-infected hosts. Now, susceptible hosts can also be infected by the vaccine at some rate beta sub v. Susceptible hosts can also be infected by the pathogen at some rate beta sub w. And then vaccine-infected and pathogen-infected hosts recover at some rates delta sub v and delta sub w, okay? Now, the sort of key assumptions in all of this is everything I'm going to talk about rests on the idea of a single well-mixed population. It's obviously a simplification, but it makes things more tractable for this first pass through the models. We're going to assume it's a perfect vaccine, so when you get infected by the vaccine and then recover, you're immune for life. There's no waning immunity. And then lastly, there's perfect cross-immunity between recovering from the pathogen, then you're immune to the pathogen, but you're also immune to the vaccine and vice versa, okay? So those are the really key simplifications here. I'm going to tweak this model a little bit in each particular section of the talk, but if you understand this basic framework, it's always going to be this same basic idea. The other thing I'm going to do today is try to focus in on a particular kind of transmissible vaccine, and that's a weakly transmissible vaccine. There's a couple reasons to focus on these, both of which really stem from the fact that if we focus on a weakly transmissible vaccine, meaning one that has an R naught less than 1, each individual infected with that vaccine will on average infect less than one other individual. What that means is that if we start, say here at time 0, and we're directly vaccinating individuals with the transmissible vaccine, it's going to spread rapidly through the population. So the proportion infected with the vaccine is going to increase rapidly, both because of direct vaccination and because they're transmitting. But we have the ability with a weakly transmitted vaccine to now say at this time point, well, we've vaccinated a sufficient amount of the population, so let's stop vaccinating directly. And because the R naught is less than 1, in principle, that vaccine should then die out. So you maintain some kind of control over the fate of the vaccine. The other reason to focus on weakly transmissible vaccines is that they're less prone to evolve. And this result really comes from a very nice paper by Rustam Antia and colleagues in 2003, where they showed that the probability of evolution in an infectious disease in general increases as a function of the pathogen R naught. And what that means in our context is the greater the vaccine R naught, the more likely it is we're going to have evolution. Okay? And obviously one of the potential outcomes of evolution in an attenuated vaccine is a return to wild type virulence, which we obviously don't want. Okay? So we're going to focus on these weakly transmissible vaccines. What I want to do now is tell you about the question number 1. Yeah, you're making me laugh about it. Okay, so the first question, right? So can vaccine transmission facilitate pathogen eradication? And the scenario we have in mind here is something like this idea of hantavirus and the reservoir population of deer mice. Okay, so hantavirus is an infectious disease that doesn't transmit from human to human, but every year some humans catch this infectious disease from wild deer mice populations. And so what you see down here is just sort of the annual incidence of that infectious disease in the United States from CDC counts. It's not a huge number of cases, 30 to 40 individuals per year. The problem is when you get hantavirus, your chance of death is somewhere like around 40 or 50%, right? So it does have real public health implications. One thing that could potentially help to eradicate hantavirus or to minimize its public health burden is to eliminate it from the wild population, to eliminate it from the reservoir population. And so what I want to ask here first is whether or not a transmissible vaccine could facilitate this eradication by lowering effectively that threshold we need to accomplish. So the basic modeling framework is going to be the same. Okay, so an SIR framework. The only differences here from the original framework I showed you is we want to model an endemic disease. So we're going to have births of host individuals, okay, and that's going to occur at this rate B. And vaccination is going to occur at birth. Okay? So when individuals are born, some fraction, sigma of them are vaccinated, and then we're also going to let there be death And this just lets us reach some kind of stable state, simplifies the math, and it allows us to focus on these kind of endemic infectious diseases. To a classical model? Okay, so I've got V as well, which is the number of hosts infected with the vaccine. So that's one extra piece. And the vaccine transmits, so vaccinated individuals infected with the vaccine infect susceptibles at that rate. Is that, got it? Okay. So yeah, it's really a classic SIR with, well, the other way to think about these models is a model of competing pathogens. That's another thing they're very similar to. The key difference then, if you want to look at the world that way, is we're adding one of the pathogens intentionally, and that's another way to think about direct vaccination. So what we were able to do with this model is essentially solve for the eradication conditions. So what does it take in terms of vaccination effort to get rid of this pathogen? To, for example, get rid of hantavirus from the deer mouse population. And reassuringly, if there is no vaccine transmission, we recover the classical result, okay? So the rate at which you need to vaccinate the population is equal to 1 minus 1 over the R naught of the pathogen, the wild type pathogen, okay? So that's good. We want to recover the classical result. What we were also able to show though is now if we let the vaccine transmit, okay, so if we model a transmissible vaccine, it modifies this classic result with this quantity shown in the shaded yellow. Okay? And what's interesting about this is, well, first you can see that it's always going to reduce the amount of effort you need to expend to eradicate the infectious disease. The other thing it shows you is that the efficacy of a transmissible vaccine, particularly a weakly transmissible vaccine for which R naught sub V is less than 1, is going to depend on the R naught of the pathogen. Specifically, as the R naught of the pathogen goes up, the percentage reduction in vaccination effort we get by transmissibility falls off, okay? So really, there are two sort of punchline results here. One is that a weakly transmissible vaccine, so one with, say, an R naught only equal to 1, it's not even self-sustaining, for a pathogen with a relatively modest R naught, so something like hantavirus, for which R naught has been estimated to be somewhere between 1 and 2, a little bit of vaccine transmission makes a huge difference. You reduce the effort required to vaccinate that population and get rid of it by almost 80%, okay? So it's a very substantial gain. Even a weakly transmissible vaccine with an R naught of only 1/2 reduces it by almost 40%. And when you're talking about vaccinating a wild population, for instance, of deer mice, this kind of thing is huge, right? Because it's not a simple thing to do. The other take-home message here, though, is that these transmissible vaccines, at least weakly transmissible vaccines, aren't going to be very effective if we use them against infectious diseases that have relatively high R naughts, okay? So maybe we've narrowed down the scope here a little bit into the kinds of infectious diseases against which these will be effective. The other thing we could do with this model is say, well, maybe you can't eradicate the infectious disease in some cases, but how much could you lower the incidence of that infectious disease within the host population? Maybe that's sufficient. Maybe you don't have to actually eradicate it. Well, what those results show is something that's really very similar, okay? In the sense that the reduction in the incidence of the infectious disease is really only substantial when you're talking about an infectious disease with a modest or relatively low R naught. So in this region here, say pathogens with an R naught less than 3, you can have a relatively large impact on the incidence of the infectious disease. So for instance, for hantavirus here and a weakly transmissible vaccine, you could reduce the incidence by, say, 30% for these particular combination of parameters, okay? So they can be effective, but the same general message holds true. If the R naught of the pathogen is too big, weakly transmissible vaccine is not likely to be an effective tool. Okay, so what I just talked about was really a question of the depth of vaccine coverage. So can we eradicate an infectious disease from a host population or suppress it? What I want to do now is turn to a slightly different question, and this is a question of rates. Whether or not vaccine transmission makes a difference when we try to combat, say, the next pandemic of influenza, okay? Or the next Ebola epidemic, or the next SARS epidemic, okay? So we took just a slightly different approach to this model. It's just a little bit simpler structurally. So because it's an epidemic model, we're not going to worry about the births of hosts or the deaths of hosts. We're going to assume the epidemic goes on a very rapid time scale relative to those processes. So we really have a very simple model. The problem, of course, is now we can't just analyze equilibria. We've got to actually ask questions about time, which is always much harder. What we wanted to do specifically in terms of these time questions, at least as a starting point, is motivated by the kind of scenario we might face with the next pandemic. And specifically, I want to motivate that by thinking a little bit about what happened with the recent epidemic of Ebola that started in West Africa in 2014. So this epidemic was observed in West Africa around March 25th of 2014, okay? This graph down here shows the days since epidemic origin and the number of infections. And what you see is right after it's observed, it starts to chug along and it's increasing. From the time that that epidemic was first identified to the first case in North America on September 30th, you've got 189 days, okay? So if you're going to try to use vaccination as a tool to control this outbreak, you've only got 189 days to vaccinate the population, okay? Sufficiently to prevent the outbreak. So a very short window of time. And based on research that's been done on, say, for instance, influenza, not even close to enough time to reach a sufficient proportion of the population. So what we wanted to do then is say, okay, well, what would vaccine transmission accomplish? Like, would it facilitate our ability to vaccinate the population in time? And to do that, we developed a perturbation approximation for the time required to protect the population. And specifically, the result I'm showing you here is essentially the fractional reduction in the time required to vaccinate the population that we get as a result of vaccine transmission, okay? So we can translate that kind of ugly expression into a nice graph that's easier to understand, which shows the percentage reduction in the time required to vaccinate the population as a function of pathogen R naught, okay? And for that example case of Ebola, which has an R naught around 2, what you can see is that we get, for even a modestly transmissible vaccine with an R naught of 1, we can reduce the time required to vaccinate by 50%, so it halves it, which is pretty striking, I think. And the same sort of qualitative result is true even for a very weakly transmissible vaccine with an R naught around 0.5. We still knock that time required to vaccinate down by about 25%. So those effects, I think, are large. And the other thing that's interesting here is because we're talking about prophylactic vaccination, the vaccine is not competing against the infectious disease itself. And as a consequence, in terms of prophylaxis, pathogen R naught actually doesn't matter that much in this case. It still matters, but it doesn't matter as much as in the other cases where you're actually competing with it with your vaccine. Now, that was a really optimistic scenario, right? That's the scenario where you see the infectious disease somewhere else and you get 189 days to respond. Obviously, that's not always going to be the case, right? We just have to switch our geographic perspective and suddenly we're in West Africa and it's not a question of we have 189 days to get the vaccine distributed. It's happening right now. And the question becomes not one of can we prevent the epidemic, but can we minimize its impact? So can we actually reduce the number of people who get sick and die? And does vaccine transmission influence that? Does it help us out in that case? Okay. So that was the second sort of way we wanted to approach this problem. And what we ultimately ended up doing here is relying more on numerical solutions. And specifically, we generalized this problem to arbitrary time points of vaccination initiation, okay? So the way to read this graph is the x-axis tells you when you started vaccinating relative to the start of the epidemic. Okay? So negative values mean you were able to anticipate this epidemic and you started some number of days in advance. 0 means you started on the day of the epidemic, and to the right means you're late. You see the epidemic and you react to it. You don't plan ahead. The y-axis shows the reduction in epidemic size. Okay? So by what percentage are we able to reduce the total number of individuals who get sick by having a transmissible vaccine? Each line is an infectious disease with a different R naught. So this brown line is something like Ebola or influenza with an R naught of 2. SARS is this blue line with an R naught around 3. And smallpox is this pink line. What you can see, hopefully, from this figure is that for a transmissible vaccine to be maximally effective, for it to have really large impacts, the sweet spot is we saw it coming, the epidemic, ahead of time, but not sufficiently far ahead, back over here, to where a traditional vaccine could have been used, okay? So the benefits relative to a standard vaccine are maximized in this sweet spot where you see it in time, but not early enough for a traditional vaccine to work. If you start trying to vaccinate past the start of the outbreak or the epidemic, a transmissible vaccine has very little positive impact. And the reason for that is at that point, this vaccine is actually competing with the infectious disease itself. That infectious disease has already started spreading. It's reducing the number of susceptibles. It's spreading immunity to the vaccine itself, and it's less effective.
I'm a little confused by the drop-off as you go
to the left.
To the left. Is that because of competition after the introduction?
Over here, why we drop back down? This is just because we're measuring it as a reduction in epidemic size caused by vaccine transmission. So it's relative to a traditional vaccine. So you get no advantage if the traditional vaccine would have worked anyway, which somewhere out here on the left, all of them would have worked because there would have been enough time.
Even if the vaccine was transmissible.
Out here, oh, crap. All that's happening is there's no benefit of vaccine transmission relative to a traditional vaccine because the traditional vaccine would have vaccinated everyone before the outbreak started anyway. So it's kind of boringly
distracting.
So that's why they go back down. Okay. So what I want to do last is talk about probably the part of this project that I find the most interesting, and that's really the consequences of evolution and also maybe some ideas about how we might control evolution in these transmissible vaccines. The way I want to structure this is by thinking about the consequences of evolution in the context of the two different kinds of transmissible vaccines, because actually I think the consequences of evolution are wildly different depending on how you've genetically engineered or developed these things in the first place. Where I'm going to start is again with an attenuated vaccine. Okay? So again, these are vaccines, you just take the wild type pathogen, you adapt it to unnatural conditions, that weakens it, you still get immunity, but if you were to go backwards in time here, you go back to the wild type pathogen. So I'm going to start by talking about those, and then I'm going to finish by talking about evolution and recombinant vector vaccines, which has very different consequences. So it turns out that we've actually conducted a really large natural experiment on evolution in attenuated transmissible vaccines. And that is the result of the vaccination program against polio that relied on oral polio vaccine. So this vaccine was developed prior to fancy genome sequencing. It was done in the old-fashioned way. You adapt it in the lab and you try it out. And you try it out until you get something that seems like it works really well. You get a good immune response, nobody gets sick, looks good, right? So, and it has been spectacularly successful. If you look at graphs of like polio incidence, once you start using oral polio vaccine instead of the previous vaccine, which was inactivated, it plunges worldwide. But what became apparent in the late 90s, early 2000s, is that as wild polio strains were becoming eradicated, we were seeing these outbreaks of polio in places where it wasn't supposed to be. For instance, in Haiti and elsewhere, okay? And it turns out that those outbreaks, once a bunch of sequencing done, were actually polio that was derived from the vaccine itself. So they're vaccine-derived polio outbreaks. And this graph down here just shows since 2000, when surveillance really started in earnest for these things, the number of cases per year. And so we're seeing these constant, repeated reversions to wild type. The reason this occurred so easily is we now know that so the oral polio vaccine was originally a trivalent vaccine, so it had 3 different strains of polio in it. And 2 of those were attenuated by genetic substitutions at just 2 to 3 positions, okay? So very little evolution is actually required to return the vaccine to wild type virulence, okay? And what was happening is that in areas where you have low vaccination coverage, so, okay, polio's gone, let's not worry about it anymore, so you lower the vaccination coverage, there's no wild type polio, so there's no natural immunity. When individuals did take the oral polio vaccine, it would get transmitted, and it would transmit in very long chains of transmission. You get evolution back to wild type virulence, okay? So this has obvious negative consequences at the individual level. If you're at the end of that chain of transmission of the vaccine-derived polio, you get polio, right? And that has very serious, very negative consequences. What we wanted to do, though, is ask, okay, obviously the case, but maybe if we're applying these to something like eliminating hantavirus in deer mice, we don't actually give a crap if any individual deer mouse gets hantavirus, right? It's just not relevant. What we care about are the population level consequences. So we pursued this by modifying these simple models to include the possibility of reversion to wild type. And we did this in a evolutionarily super naive way. We just said individuals infected with the vaccine get switched to being infected with the pathogen at some reversion rate little r, okay? And really that just changes the equations by taking some individuals out of vaccine infected and dumping them into infected with wild type. Okay, so it's a very simple model evolutionarily, but it allows us to get some insight into the population level consequences. What I'm going to show you here are the results of stochastic simulations that compare the population level consequences of a traditional vaccine up here in the first plot with transmissible vaccines. What you're looking at is the proportion of the population that's infected with the pathogen, the infectious disease. There's 5 different replicate simulation runs across time, okay? Now what's happening here is our infectious disease is at its endemic equilibrium, so it's infecting about 25 or something, 23% of the population. Here we start vaccinating, and I've just set this case up to be one where you cannot eradicate that infectious disease with a traditional vaccine. So what happens when we start vaccinating is we drop its incidence within the population, but it's still there, okay, at lower levels. What we wanted to do then is compare that to a transmissible vaccine with a low rate of reversion. Okay, so the probability or rate of reversion is 1 times 10 to the negative 4. And what you see is though, even though we have reversion, okay, like we see in the oral polio vaccine, we get a better outcome here at the population level. We've effectively eradicated that infectious disease from the population because the benefits of transmission at the population level far outweigh the few cases where we get reversion. What you can see down here, however, though, is that there's little bumps along that x-axis, and those are repeated vaccine-derived outbreaks of the infectious disease, okay? Over here, we just raise the rate of reversion, or the probability of reversion to 1 times 10 to the negative 3, and you see it's pretty obvious. It does what you'd think. This rises up a bit. So we get more vaccine-derived outbreaks, but the fact is we're still doing a lot better than we were with the traditional vaccine, at least at the population level, okay? So reversion weakens the benefits, yeah? Infectious disease? I don't know, less than 1. I don't actually remember. Sorry. Well, you mean the infectious disease? Yeah. So in this case, I don't actually remember the parameters, I'm sorry. They're in the paper, I have to look. But I mean, it's a weekly transmissible vaccine for sure. That's all we've been focusing on. I don't know, the R0 of the infectious disease is probably something like 2 or something. Yeah. So reversion weakens the benefits, but it doesn't get rid of them. What we then did is try to figure out what's a likely outcome of evolution then in recombinant vector vaccines, okay? And so again, these are those vaccines that you create by taking a vector organism, something that's transmissible within the host but innocuous, and you insert an antigenic region into it so that you have a vaccine that's transmissible. It moves from host to host and expresses that antigen of the pathogen, okay? Well, one thing that seems incredibly likely to happen in this case, so here we haven't done a big natural experiment, so we don't actually know, but based on what we know from decades of experimental evolution, if you stick a foreign gene into some organism, its first thing that's going to happen when you let it evolve on its own, it's probably either going to kick that gene out or it's going to downregulate it. In this case, that's particularly true, of course, because you're sticking into this genome a gene that makes it recognized by the human immune system, right? So there's going to be very strong selection against it. So if it mutates that form, it's going to get kicked out. Now, the potential problem here then is not at the individual level. You don't care. If I give you the vaccine, it reverts in you, and you go back to this innocuous vector, totally irrelevant to you. You'll go on, no problem. What becomes important here, though, is the population level consequence. And this is what people engineering these things hadn't really given too much thought to. And that's that, at least under sort of the simplest modeling assumptions we can make, when individuals get infected with a wild type pathogen, they then recover and they're resistant to the pathogen, but they also have some resistance to the vaccine because the vaccine is carrying an antigen of the pathogen. When an individual is infected by the vaccine and then recovers, they're immune then to the pathogen, the vector, and the vaccine. But when individuals recover from being infected with the vector itself, if that free vector is in the population because it evolved due to reversion, then those individuals are immune to the vector and the vaccine. And what you can see here is everybody's immune to the vaccine, right? So you've got competition. Once this happens, if you get reversion, then your vaccine is going to be really hard to drive through the population, okay? So what our postdoc Andrew Basinski has been working on is understanding really the conditions under which we can control that evolution, okay? When can we keep that from happening and continue to drive our transmissible vaccine through the population? What he was able to show is that you can suppress evolution in the transmissible vaccine and keep it working if the rate of direct vaccination, so this is the rate at which we introduce the transmissible vaccine into the population, if that exceeds the cost of carrying that antigenic region, multiplied by 1 minus 1 over the R naught of the vector organism, okay? So the reason this result is interesting to me is it turns sort of the conventional wisdom in designing these transmissible vaccines a bit on its head. So if you naively went out there and thought, I'm going to make a transmissible vaccine. I'm going to take a vector organism that has a wonkin har R naught, because if I do and I stick this antigenic region in it, I can just let it go and the deer mouse population is going to sweep through the whole population and vaccinate everyone. That sounds ideal, right? It would require no effort. High R naught of the vector is awesome. The problem is once you allow for evolution and reversion to the vector, and we acknowledge that there's probably some cost of carrying that antigenic region, what Andrew's result shows us is that as the cost of antigenic insert increases and the vector R naught increases, so too does the rate at which we have to directly vaccinate the population to suppress evolution of the vector, of the vaccine. So the problem is you get this sort of catch-22, whereas you increase the vector R naught, sooner or later you run into this gray region. And what that means is in order to suppress evolution of the vaccine, you have to vaccinate so much that you might as well just use a traditional vaccine, okay? So what I think this result helps to do is guide efforts to develop these recombinant vector transmissible vaccines in the sense that at least it shows that you've really got to be sort of conscious of this cost of the antigenic insert and how that might balance out with the vector R naught and the results of evolution. Okay, so just to summarize these evolutionary consequences, this is sort of how I see things shaking out at the moment. With attenuated vaccines, the problem we face is individual level risk. If something like oral polio vaccine reverts, you've got a virulent wild type pathogen. So this probably precludes ever engineering these things on purpose or attenuating them on purpose to be transmissible in the human population. However, it doesn't have really terribly negative consequences at the population level. So for things like wildlife populations, reservoir populations, it might be a viable option. Possible solutions to the problem as a whole, there's lots of talk in the literature about fancy genetic engineering to make evolution-proof attenuation. Whether that's possible or not, I don't know. I'm a bit skeptical that there's some way to just sort of stop evolution. The problems for a recombinant vector vaccine are very different. Here, individual level risk is super low. So it's not really, at least as we've envisioned, I mean, there may be possibilities we haven't considered. But in the basic sort of design here, if it kicks that antigenic region out, you just got free vector, there's no problem. The problems here, though, are more like, how do we actually implement this? Because when it does kick out that antigenic region, you've got competition with the free vector, and it seems like often suppressing that evolution, even if we can do it by swamping, in a kind of classical evolutionary sense, might be more expensive or impractical than just using a traditional vaccine. Okay? So that's how I see things right now, is each of these possible ways of engineering a transmissible vaccine has real problems we've got to grapple with. So just to sort of wrap all this up, where I think we are right now is in a place where it is very clear, I think, from the modeling work we've done so far, that the potential benefits of vaccine transmission are really potentially huge. You're not talking about 1%, 2%, 3%. So I think there really is potential merit in this idea, particularly for things like wildlife populations. And I think that's where things get pretty cool right now, is the just sheer diversity of possible applications. So there are endangered wildlife populations that are threatened by infectious disease, like great apes with Ebola or ferrets with plague. These things right now we are on the sort of cusp of developing vaccines for. But the problem is, it's not easy to deliver these vaccines to wildlife populations, threatened wildlife populations. There's been some success with baits, but a transmissible vaccine could be very effective here. The other obvious application is eradicating human infectious disease by targeting the wildlife or animal reservoirs. The other thing I think is super cool about this project is it's really sort of applied evolution in a very real way. There's like real money to be made. It really matters, right? You don't really have to sell it very hard. It's just a fact that the problems here are going to be evolutionary. And I think it's a unique sort of interesting opportunity as evolutionary biologists to really be able to sort of weigh in some meaningful and important way on something that's of relevance to public health policy. I just wanted to wrap up by acknowledging collaborators on this work. So Jim Bull has really been instrumental in driving this whole thing forward. He's doing empirical work kind of coupled with this project. Chris Ramin, who is a postdoc here at NIMBIOS, is at the University of Idaho now in the Department of Mathematics and has collaborated with me on this work, really helped out a lot. Andrew Basinski's a postdoc now in the lab working on this. Undergraduate Ryan May, graduate student Tanner Verelman, and collaboration with Rustam Antia, and then funding from NSF Beacon and NIH Midas. Thanks.
All right. Any questions for Scott? I'm just going to pass the mic around. So you can ask your question and then pass the mic to the next person.
In the attenuated evolution model where you were talking about reversion, what you described sounded a lot like when you initially described your model, it was the vaccinated individuals interacting with the susceptibles to become vaccinated. Mm-hmm. And the change in the attenuated model is vaccinated interacting with susceptibles to become infected, pathogen infected. But you didn't implement it that way. Can you go back to that?
So you're talking about, no.
Here.
Yep.
So you were including this exact step in the equations. Yep. But instead, isn't what the biology sounds like vaccine-infected host interacting with susceptibles to give a pathogen-infected host? Isn't that the biology there?
Well, I guess the biology is actually vastly more complicated than any of these cartoon versions, right? So with the oral polio vaccine, what actually happens when they look at this is you've got intra-host evolution, which obviously we're just throwing out the door, and you see the frequency of, or at least I mean, this is all done by sort of sampling stool from infected individuals or individuals who've been vaccinated. You see the frequency of the revertance is rising within individual. And then it's also then it gets transmitted to the next individual and it goes, experiences intra-host evolution as well. And slowly the frequency rises. And so this is just a super cartoon. There's many ways you could do it. We wrestled with all kinds of different ways. This just seemed like the dumbest, simplest way to do it.
That seems to be qualitatively different to me. So maybe because in that case you won't have these RV terms. Instead, in the dW by dt equation, you will have SV terms, which is not there here. So it will be something new.
Yeah, it would be totally new. I think there really are an infinity of different ways you can model reversion, and this is definitely not the right way. It was the easiest way. I don't know, maybe that's a terribly unsatisfying answer, but that's the truth.
Thanks. First of all, great talk. I really got a lot out of that. That was
Thanks.
pretty cool. I'm not so familiar with the biology for recombinant vaccines. So when you say the vector that you attach this gene to, can you just tell me why is it that you acquire immunity to the vector? Is that absolutely necessary? Is it not possible to come up with like a strain of DNA? Or something that you do?
So this is a matter of debate, and it's been incredibly confusing to me. So the people who are actually designing these things are using a vector that's called CMV. It's cytomegalovirus. It's a herpes virus. And the claim is that you can be reinfected by CMV after you've already been infected so that you don't have immunity to the vector. The problem with that is if you start reading these different papers, some people say that is true and others it's not true. And I actually don't know. I mean, I think it's quite possible that actually the truth is it's mutating or something so that you're getting reinfected, not really by the same thing. But the hope is what you said. It's just not clear that such a thing is really possible. But that is the argument of people using CMV to do this.
Thanks.
Yeah.
Hey, sort of following up on that comment, I wanted to ask about the possibility of the vector having evolution and the transmissibility. So I don't know what time scale these viruses, or if they have like this life history theory for viruses, but whether you could get, you know, in addition, like you could get like modulation, which you talked about, but if you could also get like the virus itself becoming more transmissible as
the vaccine itself,
Yeah.
like evolving greater than R naught 1. I think it's certainly possible. In the oral polio vaccine, there are different accounts of whether it has actually increased its R naught as well as returning to wild type virulence, but also become more transmissible as it evolves. But it's not really clear whether that's true or not true. But I think it's a very logical outcome of evolution
but that would be a cool thing
to look at.
to look at in the whole.
Yeah.
This is a question about costs. So you had at the end there the cost of insertion. Is there actually good evidence for anything really like a cost? And I ask this because Rustam and Bruce at Emory, they always say, well, there's no real cost to antimicrobial resistance. I mean, there's no evidence for loss of resistance. Is there something similar here, or is it completely different?
Well, so here that cost term, like mathematically, it's really the reduction in R naught of the vaccine relative to the wild type. So it could have a bazillion and one different real biological
Is there evidence
origins.
for it, though? In the
For
evidence?
real cost. Well, I guess the evidence I would argue for here is in a transmissible vaccine, there have been too few to say there's evidence or not. But in the context of inserting a gene into a foreign gene into an organism, I think we know from like 20 years of experimental evolution, don't we, that when you do that experiment in the lab, it gets whomped on by the wild type strain. So I do think that there's strong evidence for a cost of carrying some foreign genetic element. But I don't actually know they're really.
So the Beacon folks would say, oh yeah, there's good evidence for this.
The Beacon folks?
Yeah, the Beacon folks. You're part of the Beacon folks, so.
Yeah, very, very tangentially. But yeah, well, I think so. I mean, I think I don't know, like Bull's done these experiments. This is one of the great problems with genetic engineering, right? That we're starting to confront is you think you got this microbe to do what you want. You put this gene in, like, here, eat this garbage, right? And instead, it just evolves right away because carrying some foreign gene is costly. I just can't imagine there wouldn't be a cost in this case, but.
The question is how you define cost, I guess. That's
Yeah, and here it is
It's
simply the
simply
reduction
the-
in R naught.
Yeah, okay.
So, yeah.
So this is really interesting. I was just sort of wondering if you could speak to the social factor here, because I can imagine, given that there's already sort of pushback against vaccination in the developed world, how do you present the idea of a transmissible vaccine in a way that doesn't sort of set off the fearmongers and scare people? Because it seems like a very real risk in this case. You don't want, the last thing you would want would be to increase vaccine pushback.
Right. I think that's absolutely true. I have wondered a bit about this. Like, I didn't actually find all the evidence on these annual vaccinations we get being transmissible. That's stuff I uncovered recently. I just started looking, I just checked one, is it transmissible? And then it's like, if you actually ask, Google Scholar that, then you come up with these papers. Yes, it's documented, right? And you can do this for all of the vaccines. They all transmit. And I did start wondering if this is going down I feel like that information should be known, right? I mean, it's just a scientific fact that does happen. But I don't know. I don't know the right answer, right? But I do think by burying these, I guess my impression was when I found that, I'm like, well, this looks bad, right? I mean, all these things are buried and no one ever talks about it, but these things are transmissible. It's documented. I mean, are we making it look worse than it really is? It's rare, right? And in these things, transmission, it has been demonstrated to have no negative consequences. So there aren't, but I did sort of wonder if we shouldn't make it maybe more upfront that this is actually really not unusual. I don't know. Because they already exist. We've done this experiment and we're doing it all the time.
Yeah, I don't know.
Well, some of the anti-vax crazies have chicken pox. Sorry. I mean, some of the anti-vax people have chicken pox parties and other things. So they probably would like this idea. I think they object more to the preservatives that are in the vaccines, but it's an interesting question.
I think that it genetically engineered pathogen sounds scary to people too.
Oh, of course. You might trigger a different set of people.
One is anti-vaxxers teaming up with the anti-GMO crowd. Overlapping that diagram.
I just have a question. You basically have a if you're resistant to the vaccine, you're resistant to the pathogen. That's the basic assumption of your model. But I'm just wondering, what about viruses with a very high mutation rate? We talked about influenza earlier, where we have to develop a new vaccine every year, and it doesn't really work. Do you have any insights, or do you have any kind of idea?
It's surprisingly bad, actually. There's actually a great article that came out in Science or Nature a month or two ago about why it's so bad.
Yeah.
And it's actually really cool because maybe you saw, did you see this? The way they produce it is in eggs and they replicate it. And it's actually, they have the target strain, maybe they predict it perfectly, the right strain, it would have worked perfectly, but it's actually evolving in the manufacturing process to have the wrong sort of hemagglutinin sequence. It just kind of blew my mind how, applied evolution again is ruining, you got that for nothing because of evolution.
I did get it for nothing.
And so I guess the question is, to what degree if you have a vaccine in the population, so there is an additional, diversifying or negative frequency dependent selection for the pathogen to evolve something that is different from the vaccine, right? Do you have any insights into how that would adjust your results?
Well, if you play the same, so all the results, the way we've been doing everything is this sort of relativizing everything to a traditional vaccine. And so I'm not sure it would make all that much difference because, presumably that would happen with unless you imagine something like structurally different in the way we're spurring immunity and the transmissible versus traditional, the relative benefit should be the same, I think. So I guess that would be my thinking on that. Yeah.
All right. I think we're going to stop with the questions for now.